This presentation will focus on the rapid drug approval process by the United States Food and Drug Administration. In late March, many of us were glued to our TV screens waiting to hear the latest information from the Coronavirus Task Force. During these briefings, the President, Dr. Fauci or Dr. Birx, would mention the fast-tracking of new therapeutic treatments or tests for COVID-19. This presentation will discuss what it means to be fast-tracked and the possible implications, both pros and cons, of speeding up the process. FDA approval contains many steps and typically follows the process described on the slide. Step 1 is animal testing to determine if the product will be safe to start human trials. In Step 2, an investigational new drug application, or IND, is submitted. The IND will be reviewed by the IRB, which is the Institutional Review Board. The IRB is a group of scientists and professionals that will oversee the clinical research if the application is approved. This clinical research will consist of a 4-phase clinical trial. Phase 1 of the clinical trials consists of a small number of about 20 to 80 healthy volunteers. The goal of Phase 1 is to determine the safety of the product and how it is metabolized and then excreted by the body. It may last up to a year, and about 70% of the drugs will pass Phase 1. Phase 2 can have several hundred participants who have the disease or condition the drug is treating. The goal of this phase is to determine the effectiveness, dosing, and side effects of the drug. It can last several months and up to 2 years. Only about a third of the drugs will make it past Phase 2. Phase 3 is a scaled-up version of Phase 2, same goals, but expanding to up to 3,000 volunteers that have the disease or condition. It is testing for effectiveness and side effects in a more diverse population of people than done in Phase 2. About a quarter of the drugs will make it past Phase 3. And Phase 4 is the continuation of the study after FDA approval to make sure the drug continues to be safe and effective even after it is marketed to consumers. In the case of quick onset of a new disease such as COVID-19 or Ebola, or rare diseases that do not have the numbers for standard review, adjustments to the FDA approval process can occur. These differences can be a priority review designation where resources and attention towards the application with the goal of FDA action within 6 months. Drugs must prove that they will make a significant improvement in the treatment of serious conditions over the current treatments to receive this designation. The FDA assures that the quality standards for approval are not reduced, but some dispute that idea. Fast Track is designed to get drugs needed for serious conditions or that have unmet needs to the consumer quicker. It does this by providing increased access to FDA professionals and consultants on the IRB, as well as being eligible for priority review and accelerated approval. Accelerated approval will approve a drug based on a surrogate endpoint. An example of a surrogate endpoint is approving a cancer drug when it shows that it reduces the tumor size rather than waiting on clinical data on mortality rates. This will speed up the clinical trial process. And then finally, breakthrough treatments are those that treat serious conditions and improve on already available therapies. This approach gives the drug a fast track designation and path to early Phase 1 clinical trials. Although a serious condition is a judgment, all applications are reviewed to determine their qualifications for each approach. The company submitting the drug for testing can also request a particular designation. The FDA developed the Coronavirus Treatment Acceleration Program, or CTAP, in response to the urgent need for treatments of COVID-19. The goal is to provide new therapies quickly and to streamline the review and advice process to expedite clinical trials by researchers. The CTAP has already 200 drugs approved for INDs. 40 of those drugs are in the early phase, but more than 180 drugs are in late clinical phases, which include those in late Phase 2 through Phase 4 trials. Also, 2 drugs have been approved for emergency use. The therapeutics in trials can be antivirals, and these inhibit the multiplication of the virus, usually by targeting enzymes involved in the process. Also, immunomodulators. These slow down the body's immune response to the virus, which when overactive can cause the body to attack its own organs. This therapy has the most drugs in trials. Also, neutralizing antibody therapies provide antibodies from various sources, including convalescent plasma, to boost the body's ability to fight the virus. There are also cell and gene therapies in the trials. The risk of rapid drug approvals is the lack of available data or using surrogate markers to determine the safety and efficacy before the drug goes to market. Martin and Schoenfeld cite examples from Canada that show their rapid approval process resulted in 41% of the drugs being either recalled or requiring an added safety warning label compared to only 19% that went through the standard approval. They also included examples of United States FDA drugs that were approved through either fast track or accelerated approval that had fatal results. A certain leukemia drug was accelerated because it satisfied an unmet need of therapy, but was removed because of severe vascular side effects. And another drug for heart medication resulted in increased heart attacks and strokes, which led to many deaths. The quote on this slide expresses their opinion that in the long run, it is worth the time to do a complete study. In 2018, out of the 43 FDA priority approvals, 14 had breakthrough therapy status and 24 fast-track designations. Some opponents to the rapid approval believe that the FDA is serving the needs of pharmaceutical companies rather than considering the safety of the consumer. A closer look reveals the dilemma they face. Many of the novel drug therapies are for rare diseases that have limited participants in the trials. They receive priority status due to the fact that there are no other options for treatment. The idea of the fast track program is to allow experimental medications as a last resort and use data collection during treatment to assess safety and efficacy. In these cases where drugs are approved with limited study or on a surrogate endpoint, it is essential to continue assessing outcomes. Although extended clinical trials would be ideal, it would increase cost and delay access to possibly life-saving treatments. The value of rapid approval has been seen with the implementation of COVID-19 test availability. Without fast-track approval for the FDA, coronavirus testing would not have been possible, changing the target response to the disease. Due to fast-track approval and development along with the enormous number of tests that require processing, concerns about test reliability are mounting. Many companies have created tests and they have improved significantly from just a few months ago. The most common tests look for viral genetic material swabbed from either the nose or throat. Lab tests are characterized by their ability to detect a positive case, which is called sensitivity, and its ability to determine a negative case called specificity. The nucleic acid tests are searching for a specific viral RNA based on the genetic code sequence of the SARS-CoV-2. This test is highly sensitive, so if it comes back positive, it is definitive. The trouble comes with the specificity of the test. If the viral load is low, or if the sensitivity is reduced through collection, a possible false negative will result. The false negative rates are as high as 68% if tested in the first 4 days after infection, and still as high as 20% after 8 days. With such a high rate of false negative, quarantine is recommended to anyone who has been exposed or has symptoms. Treating rare conditions and responding to COVID-19 is a lesson that one-size-fits-all rules and regulations are not a reality. Triage, which is the assignment of degree of urgency, also applies to drug development and approval, proven by the FDA's adjustment of protocols. The ethical dilemma is choosing to treat with an unproven remedy or suffer losses while waiting for clinical results. Each case is different, but in this time of great uncertainty, we must be willing to take risks. Thank you.